Let's move on to the second half. Second half is a little bit more about some considerations that one must make and take when designing either preclinical research, that's research before we reach patients, and also clinical research, which is research which is done and conducted with patients themselves. So I'll keep this as brief as I can, but I think there are some important considerations for anyone embarking on that kind of research journey. So the first question is, why would you get involved in preclinical research instead of clinical research, and vice versa? So to summarize my understanding of preclinical research, I would argue that we are there really in the scientific domain, and also as doctors doing research that could be more pure science before we reach patients. Arguably, we're there to test hypotheses and elucidate mechanisms. So preclinical research tends to be the sort of more early phase work. And we may also, as I did right back in 2001, we may be developing the science and the technology that allows us to create novel therapies or novel treatments. Preclinical research with cell culture, or sometimes, of course, in animal models as well, may be available to help us consider the efficacy, so the effects of certain treatments and therapies, and also the safety of those novel technologies as well. But predominantly, we're interested in answering scientific questions. So you may have a hypothesis, and there may be a body of research literature, and that all comes together for us to answer or question things scientifically. And ultimately, you might be thinking about research that leads towards a healthcare outcome, but actually, that may not even necessarily be the case in preclinical research. Conversely, with clinical research, our objective is to assess really the impact of the preclinical work we may have done before. It's really important to engage patients in this process, and I'll come on to that in a moment. As well, I think really we're there to take what we know and has really been thoroughly developed before it gets anywhere near people, and reform our interventions or reform our therapies by doing pure clinical research. It's kind of the later stage of the preclinical work. And mainly we're answering clinical questions, i.e., does this new technology or does this new therapy help with regards to clear clinical outcomes? So, for example, does this drug lead to a greater outcome, i.e., reduce the number of cases of a certain illness, or does it lead to a reduction in mortality outcomes as well? And let's not forget, of course, there are organisations like the National Institutes of Clinical Excellence, or NICE here in the UK, which is there to help us reform guidance. And of course, we need to do clinical research in order to update our latest guidelines. In summary of that, I would argue that preclinical research is predominantly exploratory, and it grows and develops in an exploratory manner, whereas the clinical research that we do tends to be more confirmatory. And of course, these two concepts are not exclusive. There is linkage between the two, because when doing clinical research, that may raise more clinical questions that will encourage to do more preclinical work. And similarly, it's fair to say that our preclinical research can go in different directions that might lead to different types of clinical research being conducted thereafter. But clearly, the two are interconnected. Interestingly, I'd argue that the preclinical research design process is somewhat different to that for clinical research. And having experienced both, I think I can summarize that quite briefly here. So this slide was kindly borrowed by a colleague of mine, Professor Henriksson at Aalto University. And she provided this straightforward framework for the research design step-by-step process. So in preclinical research, we may start with a research plan. We may then go on to designing our experiments to really test clear hypotheses that are based on the scientific literature. And I've highlighted this in orange because this is one of the most exciting parts. I actually enjoy the experimental design because it's where we get to tailor our science. And then, of course, there's a phase of data collection and data analysis. And I find the data analysis highlighted here quite exciting because it's where you start to really figure out whether you have interesting results that may be worthy of publication in the scientific literature. And I put a little asterisk here to say, well, publication, we're moving towards an era now where everything is worthy of publication, but historically, usually it was the positive findings that were more straightforward to publish. But most importantly, in preclinical research design, it's fair to say that there will be some movement back and forth and between each of these steps. For example, every publication will raise new questions and new ideas that will take us right back to a refinement of the research plan and further experimentation. Similarly, when you're a basic scientist or an academic in the university, you might be involved in data collection and realize that actually more pilot studies are required to go and refine your experimental design. Similarly, you may analyze your data and realize that there are different models of data analysis or data interpretation that can be applied that again may have an impact on your research plan. And therefore, it's not really a linear stepwise transition through these phases. Your research path will probably involve a little bit of jumping around between these phases. And every publication, as mentioned, will lead you to consider how you could refine the previous steps in the path. Now, conversely, with clinical research, one would hope that you're not jumping around quite so much between the different phases of research. And this is because people, ethically, understandably, are a very precious commodity. We want to do everything we can ethically to do the very best for our patients and our healthy volunteers. So if we think about the similar steps, we will still have a research plan, and there will be important experimental design and phases of data collection and analysis and publication. However, I'd argue that once we get from preclinical research to the clinical research phase of any technology, it's important to make sure that we are very thorough and very robust in our steps between each phase. And that's because we really don't want to be going back to a research plan and realize that we've conducted clinical research with people that could have been better refined in the preclinical phase. So therefore, I'd argue that it's important to involve patients with patient and public involvement at a very early phase of any clinical research plan. As well, getting expert clinicians and expert scientists involved to make sure that the research plan is quite robust and that it answers a clear research question that is focused on a clear clinical need. And I think that kind of precision allows us to progress more smoothly through the subsequent phases without looking retrospectively and thinking, oh gosh, we should have changed the plan in order to optimize the study itself. Similarly, we need to be very careful in our experimental design and I'll come on to this in a bit more depth, to make sure that our ethical reviews are sound and that we are doing the very best that we can with our research design in a clinical setting to optimize our ethical considerations. When it comes to data with patients, confidentiality is of utmost importance, and it's important that we, with the hospitals that we work with, that we have very, very strong health data protection facilities and securities in place during this phase. And that's true in preclinical research as well, but it's especially important for patient-related data. And similarly, when we're doing clinical research, even before we get to the publication phase, I'd advocate registering any clinical trial that one might run on a website such as clinicaltrials.gov. That's the United States database for clinical trials that are going on internationally. It's free to do this. It's very easy to do. And it's important because regardless of the outcomes, it means that anyone can see with transparency how your clinical trial went, and that information can be useful for others designing similar or related clinical trials. Lots of information there around preclinical and clinical trial research design. Does anyone have any questions at this stage? I think we're okay. I'll continue for a little bit. That's great. So let's just give you an example of that clinical study design with the psychosis study that I mentioned earlier that I've been recently involved with. So it took over a year of planning. We had to involve a number of patients, clinicians, clinical settings, that would include outpatient settings and inpatient settings, and of course, multiple ethical reviews, because we're going to be disrupting the patient's pathway. If a patient presents with their first episode of psychosis, and then we wish to say, right, we're going to take additional brain imaging at those phases, although that brain imaging is minimally invasive, it's important to remember that we don't want to disrupt the standard of care and the treatment pathway for that patient. So we have to make sure that our study design is minimally invasive and least intrusive for those patients. So that led us to identify and recruit psychosis patients and the healthy matched controls in real time. So you don't sort of sit around waiting for 70 or 100 or however many patients you might need for your clinical trial based on your study design to all come at once. So we adopted a rolling recruitment pathway. And that's important because although a candidate for the study may come along, they may decide voluntarily to opt in or to opt out, or they may not meet the full inclusion or exclusion criteria. So we had a rolling recruitment process, which was going on actually years before I got involved in this project. What we then did was to collect both structural brain imaging data and functional magnetic resonance imaging data at baseline. And the baseline was at the first presentation of psychosis. And we had some very clear criteria on this. It had to be within a certain time frame of the first presentation. So you can imagine it can be very tricky to capture this imaging at the right time because it's in addition to what the patient would normally have. And our study design was to repeat this at 15 months and then again at 7 years, just to confirm the results I showed you earlier were related to the baseline data, the data at the time of presentation of psychosis. And we're now evaluating that data to see if there's any similar functional connectivity change at follow-up, 15 months, where patients will have had treatment and their clinical course will be different. And again, at 7 years, and I'm just to highlight, we are currently acquiring the 7-year data at the moment. So although I presented some neat and tidy results, actually, this is called the Helsinki Early Psychosis Study. That's been going on for the best part of a decade already. And only in the last few years are we able to realize important findings. And hopefully that highlights just how long it takes in order to do a clinical study in real time as a prospective study as we've designed. And therefore, it's very important to have contemporaneous, i.e. time-associated clinical records, and to invite feedback along the way, because although we want to be sure that our process is robust, if we can make little changes along the way that don't influence the entire study design, then of course we can optimize the way we do the study for the benefit of patients and clinicians as well. Briefly, I'd like to touch on ethical principles because these are absolutely fundamental to any clinical trial that you may design. And they're also important for preclinical sciences as well, particularly if work is work with research with animals in particular. So for example, the 3 R's are a great mantra to remember. So refine your methods. If you can refine your methods and use, for example, cell culture, or you can minimize and reduce the number of patients or healthy subjects you may choose in order to illustrate whatever you want to show in your study. Also, replacing your techniques, if you can do data-driven work or use artificial intelligence, then these are all the kinds of things that ethics committees expect us to do in order to reduce the burden for healthy subjects, patients, or animals in our ethical approvals. So it's important to be minimally invasive. So the least invasive we can be for our study subjects, the better, and therefore our studies will be more ethically sound and more likely to receive ethical approval. And therefore, associated with this is to be minimally disruptive. As I've already mentioned, it's important to think about the journey that the patient and the researchers will take. And we want that to be as minimally disruptive as possible so that we can respect patient and study subject autonomy as much as possible. And as well, it's important to be inclusive. So what I mean by this is that ethics committees are, in my experience, extremely approachable when one is at the very early stages of designing either preclinical work or clinical research, especially. One can engage with those committees early on. It's important to do adequate for clinical research. It's important to do adequate patient and public engagement, as mentioned earlier. And that's another reason to be inclusive, because if we do that at an early phase and realize what the considerations from the patient or study subject perspectives will be, then again, one can refine the study protocol and the plans so that the ethics committees are more likely to be happy with our research. And the way we do this in the UK is we use the IRAS system, which is the Integrated Research Application System. It's essentially a single submission portal that grants us permission to do the study. In particular, the submission that you submit will then be sent away to the Research Ethics Committee, or the REC, and they will do their reviews and have committee panels, and you may even get invited to discuss your research plans with them in order to get ethical approval. And the good thing is, in the UK, if you're thinking about designing any kind of clinical research, one must use IRAS. And although it's comprehensive. I think it's very straightforward. It also requires you to have a sponsor, and the sponsor is kind of the host organization for a clinical study. And there may be multiple sponsors if you've got a multi-site study, but these guys provide the indemnity. So they are the organizations that provide the insurances and the permissions for the research to go ahead at their location. Of course, the submission requires you to have very clear, solid, robust protocols and plans, all the relevant permissions from everyone involved, and contractual agreements between all parties. So if your clinical research involves a company, then there'll have to be clear contractual agreements between a company who might also be involved. Similarly, if you're working with a university, the university will have to have agreements signed with you and the hospital and all of the different researchers. And all of these are required in advance. So as I'm sure you can imagine, the research planning phase for clinical research, it takes a very long time, many months to get all of these documents in place, even for a very small clinical trial. But the good news is, once you have everything in place, you can use the IRAS system to consolidate that work and then take it forward for ethical review with the REC. And from there, hopefully, you get all the permissions you need so that one can safely conduct the research. And then naturally, there is an obligation to feed back to these organizations, such as the study sponsor and the Research Ethics Committee, with any outcomes. As well, if there are any difficulties during the clinical trial, one needs to report those as well. But it does make the system very transparent, and it's all about ensuring your research is conducted with integrity, which is one of my favorite philosophies. So let's summarize this. Preclinical research is often exploratory. That's more in the scientific domain, in companies or in universities, or sometimes in hospitals too. It's all about really developing new technologies and new science to help us build new clinical therapeutics. And that will be tested in the clinical research, which is often confirmatory. And like I mentioned, it's more about having a stepwise research plan and research process, which is more linear than perhaps you'll see with preclinical research. The governance and ethical requirements for clinical research, because we are using patients and healthy volunteers, human study subjects, have to be extremely tight for very good reason. And it's important to consider this right from the start, as I've mentioned, to be inclusive and to engage with the relevant organizations at an early stage. And I'd say in both preclinical research and clinical research, we really should be tailoring our research to clinical needs and clear research questions. Hopefully these have been born out of great hypotheses based on the research literature, because if we're starting with why, and we're starting with a clear clinical need, then hopefully the research is going to progress forward with adequate integrity, and one won't look back over their shoulder and think, well, hang on, this wasn't the right research to do. Instead, having a need and a hypothesis-driven approach will allow us to be very thorough and robust in our research plans. Any questions at this stage? There's a little complexity there with preclinical and clinical research. So if anyone would like any clarifications, do try me.
To be honest, Nick, I think you have been explaining everything pretty well.
Good to hear.
Well, personally, me, I don't really have any questions about the difference between those two.
That's good, that's good. And I know the audience today has a lot of experience in exploring research as well. So it's good that we're all on the same page. I've got just one last little piece to present, which is one of my favorite topics, which I call the divide. And I think it's something for all researchers and particularly academic clinicians to be aware of. The divide, as I've named it, represents the gap between preclinical research and scientific research and adoption in clinical practice. The divide is very, very large. It's something that takes a long time to cross. And I think it can be represented by this image here. So this was me recently at Aalto University in Finland as a postdoctoral researcher conducting research with some of which I haven't presented today related to medical technologies. And that was very much pure preclinical technology related research. And if I would like for that research to reach a clinical setting, I know that the path from the university to the clinical setting is going to be very, very challenging. And I know that going from bench to bedside has many peaks and troughs and is quite a treacherous journey. So if you can imagine, I think it looks a little bit like this mountain. You start off doing research and you think, well, actually, I've got a clinical idea as to why I might want to develop a new technology. But in order to move from the bench, the university environment, all the way over to clinical practice, it's going to take a long, long time. And there are many reasons for this. It's because the needs in a clinical environment are always changing. Also, what might work in a laboratory setting technologically may not be sound in a clinical setting. And often what happens, unfortunately, is that some of the research that goes on in universities isn't necessarily driven by the clinical needs, i.e. the destination. So I think it's important to remember this and know that the journey is not a straightforward linear path. In fact, I think it's fair to say that it can easily take many years, sometimes even many decades before technologies that we develop in the laboratory become close to clinical settings and patients in particular. And the vast majority of technologies and the vast majority of scientific projects don't make it. And this is especially true in drug discovery. So going back to my early research working with Pfizer as a very young scientist, I was surprised at the time at just how many drug candidates, for example, the company would work on that would never even get close to laboratory scale up or testing, never get close to cell culture work, and then never get close to perhaps animal studies, and then never get even near patients in the clinical setting because there are multiple phases to the pathway. And there are multiple phases of clinical trials. Even if a drug candidate gets as far as clinical trials, there are perhaps up to 4 phases of further trials that need to happen in order to prove safety and efficacy. So I just like all academic clinicians, especially medics doing research as well, to be aware of just how great this divide can be. So when we might have good intentions and get excited in the university setting, it's important to know that what we're working on may never reach our patients in the healthcare context. And just to summarize why this happens, why the divide exists, for the neuroscience that I've been working on, I've noticed that the complexity of the brain means that what is right for one patient may be very different for another. So it's sometimes hard to take our scientific findings and confirm or refute them in a patient cohort. As we know in medicine, we like evidence and we like to have an established consensus. So again, one study in the university setting is unlikely to change the way we practice medicine. And therefore, this means that the divide grows in time, because until we have enough research evidence to support a change in clinical practice, it's unlikely to be adopted in a clinical setting. Medical doctors, rightly, are very risk averse, myself included. And therefore, even when a new technology comes along, it may have lots of good evidence, lots of good data to support it. But many doctors and many healthcare policymakers may be reluctant to implement a new technology. There are also technical barriers, as I've mentioned. Something that works technically in a research setting may not technically be suitable in, for example, an operating theatre or in an outpatient clinic, just because the technology will be different in different locations. And as explained, evidence-based medicine is still king in Western medicine, of course. So we like to build up an evidence base before we adopt new technologies and new healthcare practices. So again, the divide is great because you need a large body of research before new policies are made or new medical approaches are adopted in clinical settings. So that's all I've got to say regarding my research journey, and as well a little bit there about the considerations for clinical trials management. I'll stop screen sharing. Thank you very much for your attention. I'm very open to your questions or discussion on the topics raised. Thank you.
Thank you, Nick. I actually have a couple of questions already, so if that's okay, I'll start.
Absolutely, it would be great.
Do you think, well, firstly, it's like a very, I don't know how you call it, philosophical question. Do you think that a lot of, for example, treatment get missed at the early stages that can potentially be successful because it never reaches up to the clinical practice?
I think you've got a good point. I think we are missing opportunities. I think it's a great shame. When you look, what tends to happen, so for example, with drug candidates, and I don't have lots of experience with drug developments, but if you look at drug candidates, they don't always have a clear pathway. So they may be deemed unsuitable for the purpose for which they're intended. But then history looks back at those particular considerations and says, well, I've just got a Siri come in there. Let's close Siri. You look back and you say, well, that candidate didn't work for that condition, but in the future, often the researchers will look back and realize, well, actually, maybe that candidate is suitable for something else. So even when a drug candidate gets lost at late phase clinical trials, it sometimes occurs that you look back and you find, oh, it's useful for other things. A good example of this would be thalidomide. So thalidomide, if you're not aware, was a drug that was developed for morning sickness. It's an antiemetic. And unfortunately, it was given to pregnant women in the 1950s, 1960s, and they had babies that unfortunately had very severe birth defects because they realized that there were two enantiomers, in fact, of thalidomide in the drug compound. And one of them was a teratogen, which was just devastating. And nobody knew this. And it was one of those horrendous situations. And of course, from that point on, thalidomide was taken out of clinical practice. It was considered unsafe to use as a drug. But then actually, several decades later, people even now are continuing to look at it and say, well, we can now split off the two isomers and we can make sure we have only the safe isomer, because it was the other isomer that was causing the birth defects. And we can use that perhaps in people who are not pregnant safely for example, cancer therapeutics. So I think you're right. I think there are occasions where drug candidates and other technologies fail early, but then they do get revisited and come on to be beneficial for the future, hopefully. So although I've explained there is a linear journey, that journey can be changed, and sometimes previous failed candidates might have a future.
Okay, that makes sense. And also wanted to ask about some practical steps of how did you yourself get involved in clinical trials, and any tips you can give as in how to actually get involved.
Yeah, that's a great question. So for me, my clinical trial experience started quite late in my research journey, having done a lot of science before even medicine. That was very much preclinical work. I did see clinical trials happening in the hospital during my PhD, but it was only after my PhD that I went to medical school. And then later still, relatively recently, I had my academic clinical fellowship as a registrar. And then I was asked by a colleague, could you help us with this technology? Could you use your 9-month research time to set up and run a clinical trial? So it was quite spontaneous. I wasn't expecting it, but I was actively seeking projects for my 9 months of research time. So what happened was, it was literally just a chance meeting between my professor and another colleague who wanted to explore the capaciflector technology that I mentioned. So for me, it was a little bit lucky, but I had also by that point had a number of research publications, and I was a registrar as a doctor, and I was a postdoctoral researcher. So I was quite sort of senior, if you like, in the research world and kind of middle grade in the clinical world. So I was in a unique position, and I had the time to embark on this. So I didn't really know what needed to be done. I didn't know much about the IRAS system. I had a good understanding of the ethical pathways, so that was fine. But I was uniquely positioned to be the chief investigator on a small clinical trial. So then I spoke to a lot of people. I spoke to the sponsoring organization was University Hospital Southampton, and that's where we ran the trial. And I basically just spoke to a lot of different researchers in the perioperative medicine research group there in intensive care and anesthetics. And I got the guidance I needed. I engaged as many different stakeholders and people at an early stage. So I would say that it all comes down to initiative. There's no substitute for experience, so I wouldn't rush into trying to run a clinical trial. I think if you're at an earlier stage of your research journey, the best way to get involved in clinical trials is to look towards clinical research where it's happening around you nearby for convenience, in a topic that interests you, and just start to align yourself with the researchers that are running that work and get involved making a small contribution. So for example, I have medical students and early stage doctors working on the data collection side. So there was actually quite a big team in this. It wasn't just me, I was running it. There was a big team of people involved. And I think therefore, if you can be one of those active participants in the team, get some experience, you'll then be in a position to have the sort of experience and knowledge you personally need to contribute to clinical trials design. And finally, I think the time is an important factor. So don't try to do this, to have too big of a role while you're at an earlier stage. For example, you're focusing on your medical school finals, or you're, practicing foundation doctor. I think it's important to take time out as an out of program research experience if you can, or to find a fellowship or an academic placement, so that there is actually protected time for this activity. Because I think to do it well, it's well worth giving yourself the time to do that. Does that answer the question, Lana?
Yeah, it does. Do you have maybe any platforms that contains like the clinical trials that are currently ongoing? Is it a thing or no?
Ask that again, sorry.
Is there any online platform, for example, where you can Google any ongoing clinical trials?
Yeah, absolutely. So I registered mine at clinicaltrials.gov, which is a great website. So you can go there, but that's a database of clinical trials all around the world. It's I think it's set up in the United States, but it's where many people running clinical trials will register. There are other clinical trials databases as well that one can look at. But I think what's important is if you're very good at data related research and you're good at handling data, then you might be able to get involved remotely, which means you can then be involved in research in your own time, which is ideal because you can fit it in around your other activities. But I would say that keeping it local is really helpful. So wherever you are as a doctor or as a medical student, you'll find that there will be clinical research ongoing. Just ask around, and you'll probably find after just 2 conversations with consultants or other academic clinicians like myself, there'll be something in your vicinity that will align with your interests, and someone somewhere will be doing either preclinical or clinical research that's related to that. So if I could be blunt, I think the reason I've had clinical experiences and preclinical research experiences over so many years now is because I've had the courage to go and look for them in places where others may not have done so. So for example, I would have a lot of conversations, impromptu conversations with different people to find the projects that I found most interesting. And you'll tend to find that both researchers and doctors doing research, whether they're basic scientists or medics, they tend to be very welcoming and receptive to kind of young blood coming through saying, I'm interested in your domain, I'd like to get involved. I think the vast majority of us are open to that. I personally don't have any active projects at this stage. I'm sort of at the writing up phase of a lot of my work, and also focusing on my clinical training in radiology right now. But there are, particularly here at Oxford and most universities, will have so much going on. Just a few conversations will get you a long way.
Yeah, in Oxford there is a lot. I've already found out from a couple of conversations everyone is involved in something.
Exactly. There's a lot of involvement. And I think one thing I would say is choose wisely. So what's really important when engaging in research? So for example, how did I choose my PhD topic and location? And people often ask me, so that you went from Cambridge University to a less familiar place in Finland. And I was like, well, actually, I met incredible people when I went for my interviews in Finland, who were exceptional professors, highly distinguished internationally. The research setting was incredible. The funding, investment, the technology available was unparalleled, world leading. It made me realize that I'm going to do my PhD studies. These are the ingredients I need. I need to be around people who I get along really well with. I need to be in a place where the technology is able to support and sustain these kinds of research projects. Lifestyle is important too, of course, when deciding where to go for a PhD. And actually, the Nordic lifestyle for me was perfect. Having been in Cambridge for a while, it was a big change, but it was a very welcome change. So I would say that finding the right people who you get along with, if you have conversations and find that the topic's interesting, but you just don't really have good chemistry with the other researchers, then maybe that's not the right project to commit to. Similarly, it may not be exactly what you want to research, but you find that you get on great with the people doing it. knowing that you're going to have good energy and it's likely to lead to something productive and some successful outcomes that you can hopefully publish. Because ultimately, it's publish or perish still. We still want to get publications to support our careers, whether that's our clinical careers, because when we apply for specialty training, we need to have publications on there to get additional credit and even to get ourselves the opportunity for interview. As well, as a researcher, pure researcher, you are very much judged for funding and for your career opportunities on your publication record. So if you've got a good list of publications in good journals, then you're more likely to able to have a sustainable career in research. So I think looking for those projects that are likely to be productive and fruitful, it's difficult to know, but if you get on well with people and you like what they're doing and they've got a good track history of publishing in that area, in that domain, then you're probably in safe hands getting involved in that kind of project.
Why did you choose Finland?
Yeah, so as mentioned, those key things about that particular research group at the AI Virtanen Institute at the University of Eastern Finland in Kuopio. And as well, I realized when I went to visit that the landscape, the lifestyle were fantastic. I went to interviews for my PhD, for PhD interviews in many parts of the world. I was finishing my master's in Cambridge. I knew I wanted to do more research in brain and technology. It's kind of what I had in mind. And I remember going to Germany, a couple of other countries for interviews and things. And I was quite keen to travel. You know, I thought PhD is a great chance to go to a different location and experience a different way of life. And I think Finland kind of chose me because when I was there for interviews and I stayed a few extra days, everything about it was just perfect. The people, the two research groups I was with, the location was just idyllic, to be honest. And the way of life wasn't quite as demanding as some of the sort of hustle bustle cities I've been working in before. It was a smaller university town. I was aware that the preclinical work I was doing was also of interest to the researchers at the university hospital there. So there's a medical school there, one of 5 medical schools in Finland is based there. So I thought, yeah, that's a good thing to have, that the preclinical research could potentially be explored in the clinical setting, like right there on campus. So that was a key ingredient. So I think there are lots and lots and lots of reasons as to why that was an ideal opportunity. The funding was also very good, and I got fortunate enough to have European Commission funding at the time, which was quite a prestigious funding package to hold. So yeah, lots of reasons, but ultimately you have to look at all of those ingredients when deciding on an opportunity. And I think I could probably give another hour's talk on how to choose a PhD project, but it's a difficult pathway. If you're not thinking about PhD and just thinking about small projects, then some of the tips I've already shared will probably be sufficient in deciding where to do research.
Okay, thank you. Maybe anyone else has questions?
I'm open to anything. You can ask me anything about my journey. I'm very transparent about these things. And if after this talk, anything crops up, feel free to contact me as well. My LinkedIn profile is probably my most active profile.
Mm-hmm.